Hello everyone, my name is Prashant Chayar Thorat. I am studying in second year B.Pharm, student of Ashokramane Institute of Pharmaceutical Science and Research, SAVE. Today discuss the subject of pharmaceutical microbiology and topic of application of cell culture in pharmaceutical industry and research. This video guided by Mr. Azad Dhage sir. Application of cell culture. Cell culture is one of the major tools used in cellular and molecular biology, providing excellent model system for studying in normal physiology and biochemistry of cell. Example: metabolic studies, aging. The effect of drug and toxic compound on the cells and mutagenesis and karyogenesis. It is also used in drug screening and development and large scale manufacturing of biological compound. For example, vaccine therapeutic proteins. The major advantages of using cells culture for any this application in the consistency and reproducibility of result that can be obtained from using a batch of clonal cells. Application of cell culture. There are many applications of cell culture. They are following. First, model pharmacy. Second, cancer research. Third, virology. Fourth, toxicity testing. 5, vaccine production. 6, replacement tissue or organ. 7, genetic counseling. 8, genetic engineering. And 10, genetic therapy. Explanation of application of cell culture. First, model system. Cell culture are used as a model system to study basic cell biology and biochemistry, to study the interaction between cells and diseases causing agents like bacteria, viruses, to study the effect of drug, to study the process of aging, and also used to study triggers for aging. Second, cancer research. The basic difference between normal cells and cancer cell can be studied using animal cell culture technique, as both cell can be cultured in laboratory. Normal cell can be converted into cancer cell by using radiation, chemicals and viruses. Thus, the mechanism of cause of cancer can be studied. Cells culture can be used to determine the effective drug for selectively destroy only cancer cells. Third, virology. Animal cell culture are used to replicate the viruses instead of animals for production of vaccines. Cell culture can be used to detect and isolate viruses and also to study growth and development cycle of viruses. It is also used to study of mode of interaction. Fourth, toxicity testing. Animal cell culture is used to study effect of new drugs, cosmetic and chemicals on cervical and growth of number of type of cell, especially liver and kidney cells. Cultured animal cell are used to determine the maximum permissibility dosage of a new drug. 5. Vaccine production. Cultured animal cells are used in the production of, of viruses, and these viruses are used to produce vaccines. For example, vaccines for deadly diseases like polio, rabies, chickenpox, measles, and hepatitis B are produced using animal cell culture. 6. Replacement tissue or organs. Animal cell culture can be used to replacement tissue or organs. For example, artificial skin can be produced using these techniques to treat patients with burns and ulcer. However, research is going on artificial organs culture such as liver, kidney, and pancreas. Organs culture techniques and research are being conducted on the both embryonic and adult stem cells culture. These cells have a capacity to differentiate into many different types of cells and organs. It is believed that by learning to control the development and differentiation of these cells may be used to treat variety of medical conditions. 7. Genetic counseling. Fetal cell culture extracted from pregnant women can be used to study the examine of abnormality of chromosomes genes using karyotyping, and this finding can be used in early detection of fetal disorders. 8. Genetic engineering. Cultured animal cell can be used to introduce new genetic material like DNA or RNA into the cell. This can be used to study the expression of a new genes and its effect on the health of a cell. Insect cells are used to produce commercially important protein by infecting them with genetically altered baculoviruses. 9. Gene therapy. Cultured animal cell can be genetically altered and can be used in gene therapy techniques. First, cells are removed from the patient lacking a functional gene or missing a functional gene. These genes are replaced